Hello, once again, I'm the Cancer Philosopher, and today we'll be talking about establishing proof of different properties of cancer. We'll first talk about some history of how these proofs came about. We'll get to what exactly makes cancer, and then we'll figure out how do we prove that something causes cancer. So first, one of the things that was being considered was cause or effect of cancer. Now, chromosomes were one of the things that were pinpointed at this time. People were considering whether or not cancer had altered the structure of chromosomes. Scientists were conducting tests with cancer cells, and they looked at the chromosomes within the cells. Chromosomes actually comes from the word chromo, which means that there's color involved here. So when they were looking at the color of these things, they saw that cancer was a little bit different. There was even some specific, in a way, mutations that were found. I don't know if they're exactly mutations, and it's not just one base that was changed, but pieces of the chromosomes look different. And what was figured out eventually, after doing some thinking about it strongly, was that there was a piece of a chromosome that was moved from one chromosome to another. And a piece of the chromosome simply moved. And that was really interesting, and it led to the theory of perhaps chromosomal changes were precipitating cancer. Furthermore, or I guess you can say on the other hand, because mutations are a little bit different, but X-rays. When X-rays were shined on cancer cells, of course X-rays would cause mutations, and X-rays would also cause cancer. So that led to the question, maybe cancer is a disease of mutations. These theories do indeed go together because they are about genes, and that's implying that cancer is something involved with the genes, and not just like black bile, like Galen had said many years earlier. Now, one of the things that happened with these theories, that was one theory. We're about to talk about another theory, but one of the things that's brought up at this point in the book is attachment to theories by scientists. And this led to to three feuding camps within cancer research. First of all, you had the virologists, who were led by Pitt and Rouse. Rouse came up with his Rouse sarcoma virus, and it was really a test of personality, because Rouse was really into his research, and he said, this is what we have to be. Viruses are causing cancer. This is what we have to focus on. And his theory really got the most attention because of that. They got the most attention because of his passion. On the other hand, you also have epidemiologists, led by two scientists named Dall and Hill, and they were more focusing on population studies. They had a lot of data, and they basically proposed that, let's say, it was caused by carcinogens, specifically smoking, for example, and even though they had the most data, people didn't focus on them as much because it wasn't as exciting. On the other hand, it was in fact the opposite. People liked Rouse's theory because it was saying that there's viruses, and you can make a vaccine to it. If you can make a vaccine towards cancer, everyone would be happy. On the other hand, smoking, if that was indeed the cause of cancer, there would only be one solution: to stop smoking, and of course no one wanted to do that. So really, something that was deciding between the camps, and the success of the camps was whether or not the population wanted to accept it. And this relates back into our understanding science class. When we have logical fallacies, and you have the appeal to an appeal to popular opinion, ad promptu populace, and people would want to take what they feel is popular, what they want to be true, and therefore they would seem to believe it. Lastly, you had a third camp. I didn't really discuss them much because they didn't really have much data at all. It was very circumstantial data, but they said that cancer was caused by genes internal to the cell. They didn't have much data at the time. People didn't really believe them, but as we'll talk about in a minute, that in fact, a little bit of it wasn't really what they were proposing, but the similar idea, thinking outside of the box, ended up being one of the major research focuses of cancer, focusing on the genes themselves and their modulation. Now, one thing that was pretty funny in the book, they talk about meetings that happened. There would always be cancer conferences and things like that where researchers would meet and talk about their research. The virologists, the epidemiologists, and the third camp would actually meet in different rooms because they didn't want to talk to each other. They hated each other so much they didn't want to see each other's research. That's pretty problematic when that happens in science because, once again, this is a lot of bias being introduced. But I found that really interesting because a lot of this is talking about what happened in understanding science. So now, like I said before, genes become something very important to focus on, and this led to the introduction of the theory of oncogenes. There's two types, proto-oncogenes and anti-oncogenes. Proto-oncogenes are normal genes within the cell itself that become activated, and when they become activated, they cause cancer. And these oncogenes are thought to be involved in normal cell cycle because cancer is, of course, an overproliferation of cells, but cells do have to proliferate in order for us to live. So you do have these genes, but basically what happens with cancer is they go on overdrive. On the other hand, you also have anti-oncogenes, and those are tumor suppressors. And what happens with them is that these tumor suppressors become inactivated, and when they're inactivated, then tumors can form. So these anti-oncogenes, on the other hand, they become inactivated. And the reason why I put the bullet point of low chance for proto-oncogenes is that proto-oncogenes, since they're used normally, you need to have essentially 2 bad copies of them. If you have, let's say, a recessive disorder, you have 2 copies of a gene, one from mom, one from dad. Both of them have to be damaged in order for the cancer to occur. Because if only you have one of them, then the dominant one can keep working. On the other hand, for anti-oncogenes, you only really need one of them to become inactivated, because then the tumor suppressor doesn't work as well. If it doesn't suppress all the tumors, then the tumor can start to grow, and that's problematic. Now, this led to a really interesting case with oncogenes. There was a scientist named Dr. Dreyer, who was also a clinician, and he had a freezer full of tumors, and that's why he was able to jumpstart into this research much earlier than everyone else. And this reminds me of an article I read in the Wall Street Journal a couple weeks ago. This was November 9th, I believe. So this was a couple weeks ago since the time of this video. But I just remembered this article, and there was this guy here, Dr. Claude Wishick, and he was talking about interactions between amyloid beta and tau, two different proteins that are thought to be involved with Alzheimer's pathogenesis. And what he did is he went around collecting brains of Alzheimer's patients after they had died. And he had these post-mortem brains, and he just took them out of the freezer and started investigating with them. Same exact things with Dreyer. Except Dreyer was even a little bit more interesting, perhaps, than Wishick over here. Dreyer had found that a piece of DNA was missing in tumor cells, and he created these probes that he kept on putting into every tumor to see if it would work. Now, the problem is that if the DNA is missing, it's not always in the same exact place, because, like I said before, chromosomes, what they found, like a piece of that is moved, it doesn't always move to the same place. The genes are not always deleted the same exact place, so it was hard to get this to work. But he kept trying and trying and trying until he could clone out that DNA. Then after he'd done that, then he would have to do the same thing in normal cells. But this led to a problem, because Dreyer was a clinician and he had all of these tumor cells, but what he didn't have was normal cells. So after he finished the first part of his research, the easy part, the normal cells, he couldn't do. He just didn't have normal cells. So there was another lab at the same time, some guy named Meier, who was working on a similar thing, and his lab was trying to find out the same thing, and they were also getting close. But Dreyer found it first, and Dreyer thought, you know what, I could try and use my little bit of normal cells, look all over the place trying to find them. It'll take a while, though, and they might beat me to it. So instead of automatically losing his case, he instead decided to set up a partnership with this other lab, with the bigger lab, and therefore they could just publish this together. Interestingly, it's once again where you get some politics in the field worked into the science, because Dreyer had to work with another lab just in order to get his theories out. Even though he had the best science, he just didn't have the resources. And that's a problem that's dominating cancer research, unfortunately. You need a lot of money to do research. You need a lot of the resources, and if you don't have the resources, that becomes a very big problem.